Good morning. I am an MSc candidate with the Animal TV Group, busy with my MSc in molecular biology, and I'm going to present some of the initial findings for my MSc. Due to habitat loss and human and animal conflict, the IUCN Red List has listed African elephant populations as vulnerable. With the fatal case of M. tuberculosis in an African elephant in Kruger National Park in 2016, as well as in the National Zoological Gardens in 2018, there's been an increase to discover what the role of disease is in these African populations. However, for TB, there is a lack of diagnostic tests, as the gold standard is a trunk wash mycobacterial culture, but due to the nature of this sample, it is difficult to culture as well as having variant results. Which brings me to the aim of my project, is to identify biomarkers to characterize immune activation in these African elephants. To do this, we focused on 4 reference genes and 7 target genes that were identified based on literature, as well as work done in Asian elephant populations. We also included some genes that were done in previous species for tuberculosis in our group. We did then amplified and sequenced these genes in order to determine whether or not we are amplifying what we think we are. And then, based on this, we tried to determine whether we could measure a difference between a stimulated and unstimulated sample using 2 types of mitogens, being the KFD mitogen, including the KFD system, as well as the popeid mitogen sample. Based on our initial findings, we discovered that popeid mitogen would be a better option to go forward and investigate the difference between stimulated and unstimulated samples. However, we saw that there was not a very big difference between non-stimulated and unstimulated samples, and thus we went on and we took 2 elephants which were opportunistically sampled and stimulated these with popeid mitogen at different time points in order to see what the optimal time point would be to characterize gene expression in these elephants. Based on this, we identified 4 target genes for further investigation, and from these target genes we identified that a 6-hour stimulation point would be optimal to measure differences in gene expression for these species. Based on this, we will now go forward and look at antigen-specific gene expression in these target genes in African elephants, and this will be the basis for the development of a gene expression assay for TB diagnosis in the future. Thank you very much.